Hello everyone, I am Abrar Rahman Abir, an undergrad student of Computer Science and Engineering from Bangladesh University of Engineering and Technology. Today I'm going to present my poster entitled Multi-Level Contrastive Learning Augmented mRNA Subcellular Localization Prediction Leveraging RNA Language Model. So what was the motivation of our study? Subcellular localization means in which compartment of cell an mRNA is located. The cellular functions, behavior, attributes are greatly impacted by the localization of mRNA. The conventional methods have considered the single-level classification to predict subcellular localization. They have not considered the multi-level classification problem. To accurately predict the multi-level classification of mRNA subcellular localization is of great importance because of its application in therapeutics, disease pathogenesis, and cellular regulation and behavior. Here I'll list the contribution of our study. At first, we have developed a novel deep learning architecture which employs multi-level contrastive learning to predict mRNA subcellular localization from sequence data. Moreover, as features, we have integrated the RNA-FM, which is an RNA language model, its embeddings, as well as the physicochemical properties. We have also integrated convolutional neural networks with multi-head attentions, residual connections, and MLP for feature discrimination. In our architecture, we have used the contrastive loss function, particularly multi-level classification contrastive loss function proposed by Vladimir et al. To enhance interclass similarity, which means in the embedded space, we push the same samples together and we push the dissimilar samples away. In this way, the contrastive loss function behaves. We have also evaluated our model rigorously against the benchmark models. For example, DM3-log, Allocator, and Clarion. As far as the dataset is concerned, we have collected the data from the RNA locate by Zhang et al. And the DM3-log by Wang et al. Here we see the distribution of the compartments and along with the percentage of RNAs located. Now I'm going to present the methodology of our approach. So here we see three different figures. The first one is the residual convolutional neural block, the second one is the encoder, and the third one is the overall architecture. Let me explain this in details. Here, at first, the input feature is passed through two consecutive convolutional neural network, which is then complemented by a residual connection, and which is then batch normalized to produce the output. So this is all about the residual convolutional neural network. So this is our encoder. The input is at first passed through three consecutive residual convolutional neural network blocks that I have mentioned earlier, and the output of these residual convolutional blocks is passed to the multi-head attention module to capture the long-range dependency and the local and the long-range dependency of the sequence. And there we have again the two residual connections, which is then followed by the adaptive average pooling to produce the outputs. So now coming to the original architecture, we see at first the input features, which are RNA-FM embeddings and the physicochemical properties, is passed through our encoder that I have described earlier. This encoder is trained with the contrastive loss function, which pushes the similar samples together and push the dissimilar samples away. And the output embedding of the encoder is then projected to an embedded space of lower dimension by a projection head. Here we have an MLP. So After training this encoder with contrastive loss function, we freeze the encoder's parameters. Then we train our classifier. Let me explain it in details. Once we have trained the encoder's output is passed to the classifier, which job is to predict the accurate subcellular localization of the mRNA. And it is trained with binary cross entropy with logit loss function as the loss function. And it produces our final predictions. So when this training is done, when this training is going on, the encoder parameters are freezed. So we can say that it's a dual test training model. Here we present our results. We have taken average precision, ranking loss, Hamming loss, and co-average as our evaluation matrices. We see that the average precision and the co is highest in comparison with other works, and in all the evaluation matrices, our approach significantly outperforms the other three benchmark models. So here is all. Thank you everyone. Goodbye.